for me to highlight. It's very important for me to highlight the essence of AMR in my perspective as an infectious diseases physician and as a physician in an endemic setting. It's not infrequent that one faces highly extensively drug-resistant pathogens in clinical isolates of patients. And when you have patients with these bacteria, such as resistant Acinetobacter baumannii or Klebsiella CPE, with almost no options for treatments, it triggers lots of questions on what should you do next and why is this happening. So as a junior researcher, the ability to ask questions is the best way to learn. And as Shlomit already mentioned, I started my way through the clinical pathway, studied medicine and specialized in internal medicine, infectious disease, and infection control. But during this time, my research interests aligned very well with my clinical practice. So I started looking into carbapenem-resistant Enterobacteriales, acquisition, colonization, infection, different infection prevention and control interventions and their impact. And then I moved to Geneva and started working on burden of AMR in European continent in high income settings, but also in low middle income settings in different projects. And when you start looking into AMR, you start facing lots of methodological challenges. And I recently received the ESCMID, the European Society of Clinical Microbiology and Infectious Diseases, young investigator grant to look at association between appropriateness of antibiotic treatment and patient outcomes. My link to One Health project started with the Darwin project in 2017, when we looked at the dynamics of AMR in urban water cycle in Europe. And nowadays we're starting a new JPI AMR project called Reservoir that I will mention shortly towards the end of my presentation. So my presentation will mainly relate to AMR burden in humans, and then AMR burden in AMR in One Health perspectives, looking into wastewater-based epidemiology of AMR and environmental aquatic reservoirs of AMR. It's impossible to talk about AMR without quoting the latest global burden study published lately in The Lancet, where they did a very large modeling exercise that yielded huge numbers of attributable and associated burden of AMR. So globally for 2019, it was estimated that almost 5 million deaths were associated with bacterial AMR, and of these 1.3 were attributable directly to bacterial AMR, with most of the burden occurring in Western and Sub-Saharan Africa. And when we talk and when we see these concepts of attributable and associated, it's important to understand that it relates to two counterfactuals. So in a scenario that all resistant infections would be replaced by susceptible infections and we don't have resistance anymore, this is the attributable, directly attributable burden due to AMR. But if the resistant infections would be eliminated totally and no infections will be occurring, this is the associated burden. And this shows us the different margins that we see in these kind of publications. So the majority of the burden was related or attributable to specific 6 pathogens, the leading pathogens here on the figure from the right side, mainly in lower respiratory tract infections, bloodstream infections, and abdominal infections. And these pathogens also align very well with the recently updated WHO bacterial priority pathogen list that was first published in 2017 to guide the research and development for antibacterials facing the AMR challenges. And nowadays in the critical group, we have carbapenem-resistant Acinetobacter baumannii and Enterobacteriales resistant to 3rd generation cephalosporins and carbapenem resistance, as well as Mycobacterium tuberculosis resistant to rifampin. And the selection of these pathogens as a critical potential and critical target and priority was due to their ability to transfer genes, resistance genes, and their global burden and the severity of infection they cause. Other pathogens such as Pseudomonas, carbapenem resistant, and MRSA were also classified as high priority due to their mainly healthcare associated burden and of specific interest in the context of One Health. VRE, vancomycin resistant Enterococcus faecium was declared as high important because of its potential to disseminate in One Health context. But when we look at these estimates and these numbers and 10 million people and 5 million people and etc., we need to understand that these are modeling studies trying to estimate the numbers, but when we try to understand the numbers that are already published in the literature, these are a little bit different, and there is a lot of data needs and data gaps in order to understand actually the burden of AMR. So in a project called Primavera, we tried to map the burden of AMR, and this project tried to predict the potential of using monoclonal antibodies and vaccines targeting AMR as alternative to antibiotics. So we tried to systematically review the published literature in order to find evidence on pathogens, infections, combinations, and outcomes, especially in important high-risk groups. And of interest in health outcomes were also mortality, but other very relevant outcomes such as disability, recurrence, need for ICU, etc., etc. And when we did this exercise, we faced huge research gaps, also in frequency of AMR, also in burden of AMR, in the health burden and the economic burden. So we could find some data on bloodstream infections due to resistant organisms, but other than that, for other infection types and for other infectious syndromes, the data was very lacking. So if we look at all-cause mortality of resistant versus susceptible infections, we find the attributable burden in the scientific literature to be very variable across different pathogens. And as you see here, it ranges from 3.4 for carbapenem-resistant Klebsiella pneumonia bloodstream infection compared to a bloodstream infection due to a susceptible pathogen, to 1.3 for Pseudomonas resistant to carbapenems. When we look at infection-related mortality, so the data is very scarce, but it's almost doubled plus the odds for mortality. But when we try to compare outcomes of patients, mainly all-cause mortality with resistant infections compared to uninfected individuals, you see that the scale of the numbers changes, and VRE is associated with 11 times higher the mortality, and MRSA with 6 times higher the mortality. But when we try to think what mediates the burden of AMR, so when a patient has a resistant bacteria causing a clinical infection, at the beginning, when prescribing the empirical treatment, while there is no identification of a pathogen and susceptibility pattern, it's not infrequent that we prescribe erroneously an inappropriate antibiotic treatment that does not give the right answer or treatment to the patient. And this inappropriate treatment is not a very rare event. So in US hospitals, 1 in every 3 patients with a bloodstream infection receives inappropriate treatment. And it has been associated with worse patient outcomes, yet this association is very hard to estimate, and it's subject to confounding and bias. And there is another factor playing in this equation, and this is the virulence of resistant bacteria. So the association between resistance and virulence in specific pathogens is very variable. It depends on different factors, such as the bacterial species. So, for example, Pseudomonas and Acinetobacter baumannii evolve very rapidly under antibiotic pressure and the virulence is increased. Sometimes the combination of a specific virulence and resistance mechanism differ. The environment or the ecological niche where the infection is happening, the anatomical site, the antibiotic concentration, and the host response, all these factors also help in this interplay. So what do we do to fight AMR? There is a burden of AMR inside hospitals, outside hospitals. As a clinician, I'm mainly interested in what's going on in hospitals. And we have different interventions, mainly infection prevention and control and antimicrobial stewardship. By antimicrobial stewardship interventions, we try to improve the prescription habits and align them with the local epidemiology. And in infection prevention and control, we try to look for multi-drug resistant organisms. We apply isolation, cohorting of patients. We do vertical and horizontal interventions, including hand hygiene, environmental hygiene, contact tracing, etc. One of the main things we use in IPC, infection prevention and control, is MDRO surveillance. So we screen for carriage of multi-drug resistant organisms, and we screen patients. We all the time collect information on these patients to try to understand and limit the spread in the hospital. And this screening, patient screening, is usually very variable in different settings. Some settings screen on admission, on discharge, weekly screening, different departments, different high-risk groups, lots of screening gaps, and it's costly and burdensome. So can we use surveillance in other targets? COVID-19 pandemic was proved that wastewater-based epidemiology is a very easy to use, cost-effective, and valid tool. It was very useful to track changes or trends in virus concentration. It helped to complement testing gaps and to evaluate populations where not many people were being tested to COVID. It enabled the evaluation of effectiveness of interventions in the large scale, and it served as an early warning system and enabled detection of new variants. But and above all that, it was quite practical and cost effective. Kashiva et al. Argued that wastewater surveillance methodology is very valid and practical, but also there are different uses and factors that are integrated in the use of wastewater surveillance. So the bottom line, we need to know what are we looking for and why in order to maximize the use of wastewater surveillance. So if we're in a pandemic context, if we're in the beginning or in the end of the pandemic, we just want to detect. And while we're in the middle of the pandemic, we want to quantify. So the sampling frequency and the prioritization of sampling population is different. So for example, while a pandemic is accelerating, we are mainly aware of the potential of overwhelming the healthcare facilities, and then testing or sampling in wastewater serving vulnerable populations such as hospitals and healthcare facilities is most important. While after the end of a pandemic, we just, or before preparing or evaluating a potential pandemic sentinel sites, such as large-scale wastewater treatment plants serving large proportion of a population should be sampled. But what about wastewater-based surveillance of AMR? As COVID really was beneficial, but what about AMR? So we ran this systematic review of literature in 2020, looking at wastewaters of hospital versus community in terms of AMR abundance. We included 37 studies up to 2017 that sampled simultaneously hospital and community wastewater, and we found that in most of these studies, resistance was higher in the wastewaters from the hospitals. This was correct also for specific subgroups of bacteria, so for CRE and for third-generation cephalosporin resistant or ESBL carrying E. coli, predominantly wastewaters from hospital were richer in AMR in these multi-drug resistant bacteria or genes compared to community. For VRE, vancomycin resistant enterococci, this was less consistent. So some studies found higher abundance in hospital, and others found more abundance in community, making this as more inconsistent with hospital contribution to AMR. Factors that we found to be affecting the hospital wastewater resistance content included the local endemicity, so what's happening in the same area, the hospital size as the size grows larger and hospitals are overcrowded, the content of the abundance of resistance is higher. There was a seasonal effect. While higher levels of ARG were released during the winter season, normalized abundance corresponding for carriage per cell were found to be larger in the summer, and the conclusion was that higher temperature allows for better dissemination of resistance and transfer of plasmids. Naturally, proximity to hospital, the proximity of the sampling point to the hospital was associated with higher abundance of resistance as well as higher levels of antibiotics. For the wastewater treatment process effect, it was variable depending on different wastewater treatment processes. One point that we found very challenging as a clinician reviewing One Health environmental AMR studies is the variation in reporting and in heterogeneity in reporting of these kind of studies. While medical research is quite standardized and there are certain rules that almost everyone applies to, here it was more variable, more heterogeneous due to the nature of the study. But then when we wanted to do the meta-analysis exercise and sum all the evidence, it proved to be very challenging. And this is how we had the Embrace Water statements. And in the statement, it was based on a consensus statement with various partners from the clinical world, but also from the from the engineering, from the One Health, from microbiology, environmental microbiology, etc., all interested in AMR in the environment. And in this list, we specify 22 statements that we recommend AMR research to follow. And these are by no means recommendations on methodology, as methods change naturally, but these are more on standardized reporting to enhance the interpretability and reproducibility of these kind of studies, simply defining the methods, the equipment that has been used, the type of sampling, the conditions of sampling, and the analysis. So we know that wastewater is abundant with resistance, and hospital wastewater is also have higher content of resistance, but does it reflect the AMR prevalence in human population? So this was a systematic review of 33 studies from over 70 countries that tried to look at two things. They tried to look at concordance between wastewater and human AMR prevalence estimates in different studies, and to check whether the methodological approaches applied in these studies were associated with optimized identification of the human wastewater association. So as you see in the right-hand figure, the different studies applied phenotypic or genotypic approaches to look at AMR in both environments. And in total, there were 24 studies that served information on both, quantitative information on both compartments, and 7 were phenotypic, and 1 provided only genotypic data, and 16 provided combined data. And when we look at these studies in total, they found very high concordance that was defined as a maximum difference of 10% between the human resistance proportions and the wastewater resistance proportions, applying both phenotypic and genotypic comparisons, and there was no significant difference between both. Upon these investigators, they advised that composite sampling of wastewater, longitudinal sampling of over 1 year time, and time and location matched sampling were associated with higher agreement. Another study in the Netherlands looked at wastewater CPE and ESBLE in 100 wastewater treatment plants serving almost 40% of the population. And they found CPE in these wastewater treatment plants in almost 90% of the plants. The most prevalent were oxalike E. coli, but also Klebsiella pneumonia, and species like Enterobacter cloacae and aeruginosa were very, very CPE carrying were very rare. In this study, they also showed that the concentration of ESBL, Enterobacterales, and CPE in the influent and effluent effluents of wastewater treatment plants were reduced with almost 2.5 log scale. They found that the CPE concentrations in untreated wastewater were positively associated with the size of the wastewater, the overall E. coli concentration, but not on the with the presence of any healthcare facility in the catchment area. Another interesting study was an experiment ran by a group of investigators that they released a known quantity of a non-pathogenic bacterium that's not always present in the environment or in clinical samples, called Staphylococcus hycus, and they tried to analyze in a certain toilet in a wastewater treatment plant catchment area, and they sampled continuously, trying to see if they can detect the same pathogen using shotgun metagenomics, and to determine the sensitivity of sampling, meaning the minimum number of sick people required or carrying people required with this specific bacterium to enable detection. And they ran the experiment twice in 2 separate days, and in duplicate samples, they were able to retrieve fragments of this specific bacterium, and they estimated detection sensitivity or a minimum number of people required for detection between 16 and 140 in the both releasing dates. So AMR is also in wastewater, and we could look for it. What about other aquatic environments? So literature is full with outbreak reports discussing multi-drug resistant bacterium in hospital or healthcare facility context, with identified potential sources in sinks, toilets, and other water sources. Usually a recurrent theme in these outbreaks is that variable success is encountered while trying to stop these outbreaks using disinfection or mitigation strategies. So what's the role of hospital environment in AMR transmission? A nice retrospective study in Germany looked into ICUs and more than 550 ICUs, and those with sinks in patient rooms, they were associated with 20% increased risk for hospital-acquired infections. So hospital sinks facilitate the spread of pathogens by promoting biofilm formation, and by using these sinks, this biofilm is disrupted, and aerosolation and splashing and contamination of surfaces occurs. There are different factors that play a role in this specific point, the sink design. So shallow basins or water flow that goes directly into the drain are associated with increased risk of contamination, and also what happens beyond the sink. So, for example, pipes from plastic are more favorable of biofilm formation than copper. Defects or changes or fluctuations in the water pressure is also associated with increased risk of biofilm formation, and misuse of sink. When patient fluids are disposed into the sinks or organic material, this encourages biofilm formation. A recent study looked at CPE prevalence in hospital environment and found in a point prevalence study, and found that the amounts of or the prevalence of CPE in water sources in the hospital were quite high, with 40% of the showers harboring CPE bacteria, and almost 20% of toilet waters. And overall, the CPE positivity rate in different samples, including different aquatic resources, was 18%, and a high prevalence up to 21% was even found in sinks and toilets and shower drains of departments where weekly descaling and chlorination is in place. One important finding in this study was the species that were identified. So Citrobacter furundi and Enterobacter cloacae were consisted almost 70% of the CPE samples, compared to less than 40% of clinical samples, hinting at the importance of these pathogens in environmental reservoirs of AMR. So what do we do? Sink disinfection, is it an option? It's something that's practiced very, very often and very variably in different settings. In Geneva University Hospital recently, my colleagues had cross-sectional study looking at the prevalence of multi-drug resistant organisms in sinks and toilets in the hospital in 26 wards. And in the point prevalence study, at least one multi-drug resistant organism was identified in 36% of the samples, and almost half of the water sources, 40% of sinks and 60% of toilets. The most frequent multi-drug resistant organism were ESBL enterobacterales, followed by Pseudomonas vim-producing. Following these findings, they ran a nested randomized trial, and sinks that were contaminated with a multi-drug resistant organism were randomized to one of three arms. All sink traps were replaced, and in one arm, chemical disinfection using sodium hyperchlorite was used, and in the second arm, thermal intervention, including steam, was used. And these interventions were applied once weekly for 4 weeks. 1 week after the last intervention, the sinks were sampled for contamination, and 3 months afterwards, they were sampled again without any intervention. And the results showed that there was no impact of either chemical disinfection or thermal disinfection compared to changing the sink trap alone, and questioned the routine chlorine-based disinfection of sinks as a practice that I must say is practiced and it's very popular, and some places even twice a day, and put chlorines, locks, different type of implementation, while this intervention is not really evidence-based and maybe not effective yet, according to this study. So how do MDRO's transmission dynamics happen within the built environment of a hospital? The frequency in which MDRO patients contaminate their environment, including the sinks and the toilets, is unclear. There are some experimental studies showing that patients with Acinetobacter baumannii carry or have a bio burden of Acinetobacter baumannii in their surfaces and in the hospital environment a little bit higher than carbapenem-resistant Enterobacterales, but the specific dissemination into aquatic environments is unclear yet. And the other direction, the transmission frequency of an MDRO from the aquatic environment back to the patient, is also unclear. There was a French study that tried to assess this transmission, looking into resistant Pseudomonas aeruginosa, and they found it to be a very rare event. But to make it more complicated, when we try to find these events, and we normally see them in outbreak contexts, less in an endemic, normal, non-outbreak context, we usually look at phenotypic similarity, and sometimes whole genome sequencing help us to look for clonal transmission patterns. But nowadays, it's recognized that also plasmid-mediated transmission of multi-drug resistant organisms is a critical transmission pathway, and normal short-read sequencing, it doesn't help us to uncover this transmission. There was a study in Singapore a few years ago that looked at the two types of transmission of CPE. They looked at almost 80% of the inpatient care given in Singapore countrywide for 5 years period. And they tried to look at the importance of plasmid-mediated and clonal-mediated transmission. The first conclusion of these authors were that the clonal transmission and plasmid-mediated transmission are equally important and should both be assessed. And over time, clonal transmission decreased with IPC, infection prevention and control implementation measures, but plasmid-mediated transmission continued to increase and was not affected by implementation of interventions aiming to stop.
Just mute everybody.
Thank you. Aiming to stop the transmission. Another important finding was that while clonal transmission decreased, and that with associated with direct ward contact, so patients were in the same ward in the same time, and there was a direct link. Other clonal transmission events and plasmid assumed mediated transmission events were not associated with indirect or hospital direct or indirect transmission, meaning that these transmission patterns were not affected by IPC measures, and there might be persistent reservoirs in the hospital feeding these transmission events over time. And a nice finding from the study was that when they looked at clusters of CPE countrywide, so the largest time span of a cluster, looking at the first and the last isolated on the time scale, when in clonally transmitted isolates were 100 days. For plasmid-mediated transmission, it was more than 670 days, hinting at the persistence of these plasmids in the environment beyond for an extended period of time. And this brings me to reservoir. This is a new JPI AMR project that we're involved in here in Geneva University Hospital with other partners, that we're going to look at aquatic environments and wastewater surveillance in hospitals for antimicrobial resistance. So in this project, we're going to concentrate on two specific bacteria, Carbapenem producing Enterobacterales and ESBL Enterobacterales, excluding E. coli. And the thought of excluding E. coli is that we believe that this bacterium is also prevalent in the community, and we're interested more in the hospital setting. On board, we have great partners, 4 clinical sites from Geneva, Besançon in France, Rambam Hospital in Haifa, and Monash University Hospital in Melbourne. And we also have Institut Pasteur and Belgium, Cinsano and University of Liège. It's a prospective cohort study that will run on 9 months, in which we will include routine patient screening in selected study wards for looking for CPE and ESBL Enterobacterales. And simultaneously, we're going to sample sinks and toilets and wastewaters to look for the transmission dynamics between the different compartments. We're going to run 2 types of analysis. We're going to have an epidemiological analysis looking into patient-related factors, ward-related factors, and hospital-related factors. And we're going to have sequencing, including short and long-read sequencing, to try and establish the transmission dynamics by evaluating clonal transmission and plasmid-mediated transmission, and to understand the frequency and the prevalence and how these CPE and ESBLE interplay between patients and hospital aquatic environment. At the end, we're going to have the epidemiological data and the sequencing data fed up into a modeling work by Institut Pasteur to try to establish all of the dynamics. So at the end, we're going to try to test and validate a sampling strategy for hospital aquatic environments, sinks and toilets, for surveillance of AMR in the hospitals, as well as sampling of hospital wastewaters for sentinel surveillance of AMR of these bacteria. We're going to try to find out what's the frequency of transmission between patients to the aquatic environment and to the hospital wastewater, and from patient room aquatic environment back to the patient, both by plasmid-mediated transmission and clonal transmission. And we're going to try to also to check what bacterial factors are influencing the persistence of certain resistant bacteria in the aquatic environment, in the near aquatic environment, in patients' rooms, sinks, and as well as in hospital wastewaters. Then we're going to try to look at the impact of patient, ward, and hospital level factors on the transmission dynamics between the different compartments, and suggest a modeling framework to analyze the transmission dynamics between these compartments. So this is what we are planning. In reality, I know, and everyone knows, that other obstacles and challenges are on the way. And as in research, if anything can go wrong, it will go wrong, and you will need to think and to see how you overcome obstacles and find creative solutions in order to achieve your goal. And this, I think, what makes research in general, and also in One Health field, more interesting, when you are trying to bring multidisciplinary teams from different backgrounds interested in the same topic and having the same goal, to communicate with each other and to work with each other when the nomenclature may be different, or the ways used may be different. But at the end, the impact of such collaboration might be more influential to both clinical medicine, but as well as human genera and One Health of global health in general. And here I would like to thank my mentors along the way, Professor Michael Paul from Rambam Hospital, and Professor Stefan Harbert and Dr. Marlike de Kraker from the Geneva University Infection Prevention and Control team, and people I was blessed to work with in my research journey in Primavera project and in Darwin and in the upcoming Reservoir project. Thank you very much.
Thank you very much, Nasreen, for a very important and interesting talk. Just before we continue, I want to remind that we have another 2 webinars in July 31st with Professor Andreas Thiemann on the SAR project with water quality. And then on August 14 with Eitan Sessler, who about the business side of aquatic pollutants and antibiotic resistance. So we hope you join us also on the next webinar. And now please, whoever wants to ask any question, open your microphones, open your camera so we can see who's talking, and we are open for questions. It said that we switched to Swedish, but we are not. Any questions so far? No, so I would like to ask questions. So do you see any change in policies in the European community toward dealing or coping with the antibiotic resistance?
Yes, of course. Even in the short time span of our first JPI AMR project, and now I feel the difference and I see the difference, although there has been always interest, but I feel that the COVID pandemic has boosted this interest in general in understanding the potential of applying One Health tools in order to understand and to drive the policy fighting against health threats and AMR specifically. And as we see how challenging is the AMR field of research in terms of evaluating trends of resistance in populations and how biased the information that we do get, because they mainly come from healthcare facilities, hospitals, hospitalized patients, and they do not reflect the overall burden in the community level. You see the potential of these kind of research and findings to enhance our understanding and maybe potentially get to more effective interventions. But we're still very far away from what we could have achieved from this type of research. And I think that as a clinician and clinical from clinical fields, they should be encouraged to reach out to other people involved in One Health research, and the opposite is correct. So teams that are working on One Health questions should also try to reach out to clinicians early in the process of working on such research in order to maximize the benefit and the potential of such research.
Thank you. And maybe one private question is, why did you choose this path in your career? I mean, you did your MD, so why did you go to research, and why did you choose this antibiotic resistance?
Well, I guess as a clinician, you get a lot of freedom to choose along the way. So I think I fell in love with infectious diseases during my clinical studies, even before I started training. But when you work in infectious diseases, it's a nice field of research and practice, because you're dealing also with other living compartment that's non-human, and the interaction between people and bacteria is always there in the background. And when you get the challenges of AMR and you see that you see the bad prognosis of AMR in the global scale, and how pertinent are these questions, and to do better, it just gets you into more interest to try and understand and maybe give better care in the future.
Thank you very much again. And I don't see any more questions. So thank you very much again for the talk.
Thank you.
Hey, Nazreen.
Oh, sorry. Yes, there is a question.
Miriam from Spain.
Hello.
I would like to ask you, thanks very much for this fantastic talk. I would like to ask you, after your review, do you recommend some kind of sink disinfection technique, or none of them are useful?
Well, it's a very hot topic that nobody really knows up to date what is the golden solution to this problem. Meanwhile, in the highest level of evidence was done in this randomized small scale study here in Geneva, and it proved no difference. So it looks like the solution is not actually the disinfection of the sinks, it's more the behavior around the sinks. So there are lots of works, for example, taking out sinks from ICUs and showing possible beneficial effect, or restricting the use for sinks for only hand hygiene, or trying to do some kind of design solutions to make the water sources more away from the patients. In our reservoir project, we're not going to do any intervention. It's going to be observational only, but the plan is that we're going to collect also data on these specific practice. So what disinfection is used, and how often is it used, and what is the architecture of the specific ward we're studying. So I believe that we're going to have some kind of more data to understand the context. But meanwhile, I haven't found any very successful strategy to disinfect sinks, unfortunately.
Okay, thank you very much.
Thank you.
Amazing. Well, thank you. Any more questions? Okay, so thank you again.
Thank you very much.
Have a good day.
Thank you.
Bye.
Bye-bye.